So we are here. We are here on the chapter number 6, the anatomy of flowering plants. So we are going to study the tissue system. First part, context. The second one is the anatomy of dicotyledonous and monocotyledonous plants. Okay. Now, so what's in the introduction? Let me see. Okay, this chapter introduces you to the internal structure and functional organization of higher plants. Right. Study of internal structure of plants is called anatomy. It says plants, right? Plants have cells as the basic unit. Cells are organized into tissues, and in turn the tissues are organized into organs, right? Different organs in a plant show differences in their internal structure. Within angiosperms, the monocots and dicots are also seen to be anatomically different. Internal structure also show adaptations to diverse environments, right? So this is the anatomy of flowering plants. The first context is the tissue system. Now, what about the tissue system? Like types of tissues based on the types of cells present, right? How tissues vary depending on their location in the plant body. Tissue depends on the locations. Their structure and function would also be dependent on the location. So, okay, their structure, function also depend on their location, and on the basis of their structure and location, right? There are three types of tissue systems. The first one is the epidermal tissue system, basis on the structure and location. The second one is ground or fundamental tissue system, another name, and the vascular or conducting tissue system. It contains the phloem and xylem and cambium, right? Now, first context is the epidermal tissue system. Now, in the epidermal tissue system, it forms the outermost covering of the whole plant body and comprises epidermal cells, stomata, epidermal appendages. Epidermal appendages are trichomes and hairs. The epidermis is the outermost layer of the primary plant body. It says the primary plant body, right? Made up of elongated, compactly arranged cells which forms continuous layer. It is continuous. Epidermis is usually single layered. It is single layered. Epidermal cells are parenchymatous with small amount of cytoplasm lining the cell wall and a large vacuole. So epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. They have the large vacuole, right? Important. The outside of the epidermis is often covered with a waxy thick layer called the cuticle, which prevents the water loss. The cuticle is absent in roots. Stomata are structures present in epidermis of leaves. Stomata regulate process of transpiration and gaseous exchange. What does stomata does is the transpiration and gaseous exchange regulation. Now what is the stomata? Now stomata is composed of two bean shaped cells known as the guard cells, which enclose the stomatal pore. So stomata is made up of two guard cells and has the stomatal pore. So this is the stomata, and it regulates the transpiration and gaseous exchange, right? In grasses, the guard cells are dumbbell shaped, right? The outer walls of the guard cells away from the stomatal pore are thin, and inner walls, that is towards the stomatal pore, are highly thickened, right? The guard cell possess chloroplast and regulate the opening and closing of stomata. Sometimes a few, not every time, but sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as subsidiary cells. The stomatal aperture, guard cells, and the surrounding subsidiary cells. The stomatal aperture, guard cell, and surrounding subsidiary cells are together called stomatal apparatus. Like, so this is the stomatal apparatus made up of subsidiary cells, guard cells, and then stomatal pore, that is aperture. Now first diagram is of the grasses. What? Dumbbell shaped, right? Dumbbell shape, right? Grasses show the dumbbell shape, right? And double dumbbell shape stomata with the guard cells, that is the okay. Am I something missing, like stomata with double double. No, stomata with dumbbell shape. Okay, no. Wait. I'm okay. So in the first diagram, stomata with bean shaped. Grasses have the bean shaped, right? Grasses don't have the bean shaped. In grasses, the guard cells are dumbbell. Okay, so the second diagram is of dumbbell shaped, means in grasses, right? The second diagram is of the grasses, and normally stomata with a bean shaped guard cells, right? Normally it is a bean shaped, but in grasses it is a dumbbell shaped. Don't get confused, right? The cells of epidermis bear a number of hairs, right? The root hairs are unicellular elongations of the epidermal cells and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. So in roots there are root hairs, in stem there are trichomes. What does trichomes? They are the in the shoot system are usually multicellular, maybe branch or unbranched, and stop and steep, even be secretory. Trichomes help in preventing water loss due to the transpiration. Now the second system is the ground tissue system. Now the ground tissue system, what is its all tissues except epidermis and vascular bundles constitute the ground tissues. It consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma, right? Parenchymal cells are usually present in cortex, pericycle, pith, and medullary rays. Okay, in the primary stems and roots, right? In leaves, the ground tissue consists of a thin wall chloroplast containing cells and is called the mesophyll. So mesophyll cells present in the leaves, and parenchymal cells present in cortex, pericycle, pith, medullary rays in the primary stems and leaves, right? So this is the ground tissue system, right? Now the third system is the vascular tissue system. The vascular tissue system consists of complex tissues. The phloem and the xylem. The xylem and the phloem together constitute the vascular bundles, right? In dicot stem, cambium is present between the phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form the secondary xylem and phloem tissues, and hence are called the open, because they are open to the secondary growth. While in the presence of the cambium, so open vascular bundles. In the monocot, so it was in dicot. Now when in the monocot, the vascular bundles have no cambium present in them, so this is why they are closed. They do not form the secondary tissues. They are referred to as closed. When the xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called the radial, as in the roots, right? As in the roots. So you can see in the first diagram, that is xylem and phloem, they are arranged in a different radii. When the xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, right? The arrangement is called radial, such as in roots. In conjoined type of vascular bundles, the xylem and phloem are jointly situated. Xylem and phloem, they are jointly situated along the same radius of the vascular bundles, right? So these are the in conjoint, right? They are at the same radius, right? Such vascular bundles are common in the stems and leaves, right? The conjoint vascular bundles usually have the phloem located only on the outer side of the xylem. Usually they have the phloem on the outer xylem. You can see in the diagram, phloem is on the outer side, then the cambium, and then inside the xylem. So this is various types of vascular bundles. First one is the radial, second one is the conjoint closed, and third one is the conjoint open. Okay, now anatomy of dicotyledonous and monocotyledonous plants, the second context of this chapter. Okay, we study the transverse section. Now dicot root. What are the dicot's root? So dicot root. So the outermost layer is epiblema. Firstly, epiblema is the outer layer. Many of the cells of epiblema protrude in the form of unicellular root hairs, right? The cortex consists of several layers of thin wall parenchyma cells with intercellular spaces. The innermost level of cortex is called the endodermis. Inner to the cortex there is endodermis, and then there is pericycle, then there is protoxylem, then there is metaxylem, then there is phloem, and then there is pith. So in endodermis, a single layer of barrel shaped cells without any intercellular spaces. Okay, the tangential as well as radial walls of the endodermal cells have a deposition of water impermeable waxy material suberin in the form of Casparian strips. Okay, they hinder the water flow. Right. Next to the endodermis lies a few layers of thick wall parenchymal cells referred to as pericycle as a tail. Now in here pericycle, in the dicot root, initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells, right? Okay, the pith is small and inconspicuous. The parenchyma cells which lie between the xylem and the phloem are called conjunctive tissue. The parenchyma cells which lie between the xylem and the phloem are called the conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissues on the inner side of the endodermis, such as pericycle, vascular bundle, and pith, constitute the stele. So this is it. So all inner to the endodermis, such as pericycle, pith, and vascular bundle, they constitute the stele. Now talking about the monocot root, right? You can see in the diagram root. You can see monocot root, right? The anatomy of monocot root is similar to the dicot root in many aspects, but it has epidermis, cortex, endodermis, pericycle, vascular bundle, and pith. So this is the sequence: epidermis, cortex, then endodermis, then pericycle, then vascular bundle, then pith. But as compared to the dicot root, which have a fewer xylem model, that is what it is, like 2 to 4 xylem and phloem patches, right? 2 to 4, where it is, yeah, it's written 2 to 4 xylem and phloem patches. But as compared to dicot root, which have fewer xylem bundles, there are usually more than 6, that is poly or xylem bundles in the monocot root. Pith is large and well developed. Monocot roots do not undergo any secondary growth. So this is it. So root here, epidermis, then cortex, endodermis, pericycle, then there is phloem, protoxylem, then pith, and metaxylem in all these things in the diagram. So first diagram is of the dicot root, and the second one is the monocot root. All this is the primary. Okay, now dicot stem. So they also have the epidermis, the outermost protective layer of the stem. Then there is a cuticle. It may be a trichomes and few stomata. Multiple layers between the epidermis. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. Okay, the cortex, it is between the epidermis and pericycle. It consists of 3 subzones. The outer is the hypodermis, consists of few layers of collenchymatous cells just below the epidermis, which provides the mechanical strength to the young stem. So this is the hypodermis, collenchymatous cells, and strength to the young stem. Then cortical layers below the hypodermis. It means below the hypodermis. Below the hypodermis consists of a rounded thin-walled parenchymatous cell with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. Inner layer of the cortex is the endodermis. The cells of the endodermis are rich in starch grains. In roots, what we study, there is a Casparian strips of suberane waxy material, right, impermeable to water. Now, innermost layer of the cortex is called endodermis. The cells of the endodermis are rich in starch grains, and the layer is also referred to as starch sheath. It is starch sheath, and there was a Casparian strip, right? Now, pericycle. Now, pericycle is present on the inner side of the endodermis. Inner side of the endodermis, there is a pericycle present, and above the phloem in the form of semilunar patches of the sclerenchymatous. And above the phloem in the form of semilunar patches of the sclerenchymatous. Okay, I repeat it again. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of the sclerenchyma. Like in between the vascular bundles, there are few layers of readily placed parenchymatous cells which constitute medullary rays. Okay, a large number of vascular bundles are arranged in a ring, as you can see in the diagram. It is kind of ring. Okay, it is kind of ring. Vascular bundles are present in the kind of ring. A ring arrangement of vascular bundle is a characteristics of dicot stem. Okay, now each vascular bundle is conjoined and open with enlarged protoplasm. Okay, we study vascular bundles is conjoined. That is conjoined, it means in stem and leaves they have the conjoined type of vascular bundle. Now it is open because it is for dicot. If it's monocot, it is then closed with enlarged protoplasm. That is primary protoplasm form first. With enlarged protoplasm, a large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. Now you can see in here a large portion. This is the pith, white in color. Okay, now see the diagram. You can see first is the epidermal hairs, then epidermis, then epidermis, then hypodermis, then parenchymatous, then parenchyma, that is pericycle, then inner no, then endodermis, then inner to it, that is the pericycle. Okay, patches of in the semilunar patches of sclerenchyma, right? And then vascular bundles, they are the ring type of arrangement, then medullary rays, and these are the then pith. So this is the diagram of the dicot. Now the monocot, we are going to study the monocot, right? They were the similar patches of the sclerenchyma, right? Now the monocot stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath. Each surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue, right? A large conspicuous parenchymatous ground tissue. The vascular bundles are conjoined and closed, right? Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water-containing cavities are present within the vascular bundles, as you can see this diagram. Okay, the ground tissue is conspicuous. The ground tissue is conspicuous. Parenchyma is ground tissue, and it is not further divided, so it is undifferentiated type of things, right? Epidermis, hypodermis, very simple. Then smaller vascular at the periphery, and central being the large vascular bundle, they containing the water cavities, and then there is a ground tissue. So this is monocot also. This is monocot stem. You can see first epidermis, then below it, that is hypodermis, then vascular bundles, then phloem, then xylem, and then ground tissue, right? So this is it. Now we are talking about the dorsiventral lip, that is dicotyledonous lip. Okay, there is also the vertical section of dorsiventral lip through the lamina shows three main parts, namely epidermis, mesophyll, and vascular system. Same three parts: epidermis, mesophyll, and vascular system. Okay, because ground tissue system is mostly the mesophyll cells in the leaves, right? And vascular system, that is third system. The epidermis, which covers both the upper surface, that is adaxial, upper surface is adaxial, that is adaxial epidermis, and the lower surface, that is abaxial epidermis, okay, of the lip, has a conspicuous cuticle, and lip has a conspicuous cuticle. The abaxial epidermis generally bears more stomata, that is the abaxial, that is lower surface. The abaxial, it's kind of opposite, you can remember like that. It's kind of opposite, that abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. That means talking about the adaxial, that is upper surface, may even lack. It's completely opposite, right? The tissues between the upper and the lower epidermis is called the mesophyll. Now the tissues between the upper and lower epidermis is called the mesophyll. As we are talking about the ground tissue system, mesophyll, which possesses chloroplast, carry out photosynthesis, is made up of parenchyma, right? It has two types of cells, the palisade parenchyma and the spongy parenchyma. Okay, the axially placed palisade parenchyma, that is on the upper side, is made up of elongated cells which are vertically, not horizontally, which are vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extend to the lower epidermis, that is abaxial surface, right? There are numerous large spaces and airway cavities between these cells. You can see substomatal cavity. Okay, now the vascular system. Now the third type of system, that is vascular system, includes vascular bundles which can be seen in the veins and the midribs, right? The size of the vascular bundles are depend on the size of the veins. The bigger the vein, the bigger the vascular bundles. The veins vary in thickness in the reticulate venation of the dicot leaves. They have the reticulate venation. Monocot leaves have the parallel venation. So in reticulate venation there is a veins that varies in thickness, so the vascular bundles too. The vascular bundles are surrounded by a layer of thick wall bundle sheath cells. The vascular bundles are surrounded by a layer of thick wall bundle sheath cells in the dicot leaves, right? Look at the figure and find the position of xylem in the vascular bundle, right? In leaves I think it is, I don't know, but anyways, isobilateral, that is monocotyledonous leaf. Dorsiventral, that is dicotyledonous leaf. Okay, the anatomy of isobilateral leaf is similar to that of dorsiventral leaf. in many ways. It shows the following characteristic differences. In an isobatter leaf, the stomata are present on both the surfaces of the epidermis. The stomata are present on both the surface of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. As you can see in the diagram, that it's not differentiated into palisade or palisade or spongy parenchyma. It's just the mesophyll, as you can see in here. So as you can see in here, okay, now we are talking about the grasses. Grasses also a monocot. In grasses, certain adaxial epidermal cells, it means the upper part of the leaves along the veins, modify themselves into large empty colorless shells. These are called the bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. And when they are flaccid means due to water trace, they just get shrink and they make the leaves curl inward to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles as seen in the vertical section of the leaves. So it's not it's only in the reticular venation that varies, but in parallel venation the size of the veins are same, so the vascular bundles are also the same. So this is the isobilateral, that is monocotyledonous leaf. Now we look at the diagrams. First diagram of dicot leaf, starting from like adaxial surface, that is upper surface, lower surface, that is apexial surface, right? Then there is at the apexial surface there is stoma, then there is substomatal cavity, then above that there is spongy mesophyll. They have some intercellular spaces, then air cavity is present, then palisade, that is vertically and parallel attached arising from the adaxial epidermis, and then there is bundle sheath, xylem and phloem. Xylem on the outer side, phloem on the inner side, right? So this is the dicot leaf. And then talking about the monocot leaf, adaxial epidermis on the upper surface, then there is mesophills on the side in the middle, that is xylem. Then there is substomatal cavity, then apexial surface epidermis, that is downside, and then there is stoma, and then there is a phloem in the like below the xylem. So this is it. Now I think now the summary is left.